Can you hear us now?
Yeah.
Can you?
Yeah.
On the computer? All right. Ha. Okay, we are all set then. Okay, let's just get this going, this eye candy. Then we'll start very soon in about 3 minutes. Okay, let's set things up. Okay. Two more minutes. 2 more minutes to get started. Okay. All right. So what you need to do is you want to turn this around. This is the button. Yeah. 1 second. Wait. Hold on. Hold on. Hold it. Just making sure that this camera is set right. I'm not looking too fat. Then we get going. Okay. All right. 3, 2, 1. Hello.
Namaste.
Vanakkam.
Bonjour.
Hola.
Ciao.
Marhaba.
Jumbo.
Ni hao.
Welcome, fellow Foldscopers, to the third live Foldscope workshop.
My name is Lakshminarayan Iyer. I am a staff scientist at the National Institutes of Health. I'm a computational biologist, so most of my work time, I spend in front of a computer analyzing various aspects of life. But in my free time, among the many things I do, I spend a lot of time looking at microbial life.
My name is Dr. AI, and I love looking at organisms under a microscope.
You do?
Yes.
Okay, here's a question for you. How many species do you think there are on Earth?
Of life?
Yes.
6 million.
6 million. Some people said 8 million. It could be up to 100 million. We really have no idea. And you know what's the funny thing?
What?
99% of life is microscopic. That means what you see around you, right, is just about 1%.
Really?
Yeah. It's just 1% of life that we see around us. 99% of life, diverse life, not the same type as we see with our eyes, is microscopic. So if we just use our eyes, we are really not looking at the whole living world. Now, there's another very interesting point and that is the basic theme of today's workshop. And that is that over the years, when people started comparing life forms, they started looking at the DNA sequences and so on, they found that life has a common origin. All life is actually made from the same set of cells. There was a very ancient cell from which life forms sprung around. What does that mean, You're related to me, right? All the humans watching us on YouTube today are related to us by, say, maybe, it depends. Some people are related to us by within 100 years, some within 1,000, some within 10,000. That's normal. All humans are related. Now you see that plant right there. You see that plant?
Yes.
You are related to that plant too.
Really?
Yeah, you are. You see the tardigrade down there?
Yeah.
You are related to that too. But the number of generations by which you're related is very far. The tardigrade, I could guess maybe about 650 million years, maybe about 1 billion years. The plant, maybe a little more than 1 billion years, right? So life has been around for a very long time on Earth, about 3 billion years. And when you start comparing all the different life forms, here's what you start looking at them, you wonder, and then you put it all together, and what people found was that life can be represented as a beautiful tree. It's almost a gigantic tree with many branches, with relations between different organisms shown as branches. So let's try to look up the tree of life. Now just hold on. You can sit here. Right? If you look up the tree of life, that's fine. So, very long time back, 3 billion years back, there were many cells that were competing with each other. One of them gave rise to what we call as life today. The others, nobody knows what happened to them. Now, this life gave rise to 2 primary forms. I would say, this is just a guess, by about 3 billion years, there were 2 basic forms of life. The archaea and the bacteria. I'm writing in mirror. Okay, now, for a long time, life was about these 2 types of organisms. They formed many different branches, many different branches, many different types of bacteria, many different types of archaea. And then, at some point, a magic event happened. Have you heard of the nucleus?
No.
Cells. Have you looked at an onion cell? Now, the center part was nice and dark blue because we had changed the nucleus, right?
Yes.
So now that part of the nucleus, right, that is where the DNA is in some cells. Now, when you look at bacteria and archaea, the DNA is not inside a compartment. But then at some point, there arose this form, like us, called as the eukaryotes. This one. And it was a magic event because in reality these were formed when a bacterium of a particular type combined with an archaeon of a particular type at the base like this to give rise to the eukaryotes with a complex nucleus, complex endoplasmic reticulum, and many things that we enjoy looking at under a foldscope. But this is a foldscope about as many forms as possible about life. Now, archaea, they are you can get them. They are found in extreme conditions. They're also found in the rumen of a cow. But unfortunately, I don't have any archaea today. But bacteria, I have quite a few. And so now let me get myself set up, right? And you get set up, Dr. AI, with the phone, and then we go ahead from there, okay? All right. So we don't need the stand anymore, right? Okay. So turn it around, okay? And then turn this around, okay? So that your finger doesn't fall. All right. So now let's get started with the fun stuff. Today, I'm going to show you many different forms of life that I grow on this bench, and perhaps that. I have no more space than this much to grow my stuff. I love plants, I love growing plants, but you can grow 1 million different life forms just on your bench. And it's fascinating. All right, so now let me show you what I have. We started off, I don't know if you were there from the very beginning, we had this spiderwort that is very special for Manu. And I actually, after I heard this from Manu, I got the plant maybe about 4-5 years back. I planted it outside, and I can show you maybe later. I have 100 flowers of spiderworts. And every time I see this, I think of Manu and all the people who've looked at the beautiful trichomes of the spiderwort under a foldscope. All right, so now let's get this moving. I have a mix of things today. The first thing is that I want to show you all some things that I've prepared in the sense. Since I have a lot of life forms and it takes a little bit of time, I have some things that I've prepared for you all, and I have some things that I will prepare to show you how things are done. All right, so now let's begin with bacteria. Bacteria are an amazing life form. They're everywhere. Here's a trick question, actually here's a question. How many cells do you think your body has, Dr. AI?
1 million?
Million is a bit less. Guess again. Okay. So let's go back to our question. How many cells do you think our body has?
1 million.
It's not a bad guess, but in reality, the number is way more than that. There are 30 trillion cells in our body. How many?
30 trillion.
30 trillion cells in our body. And do you know how many of these 30 trillion cells are... okay, so now we also have bacteria in us and archaea in us too. How many bacteria and archaea in us do we have, you think?
1 billion.
No, 30 trillion. We have the same number of bacteria all over our cells as much as we have our own cells. 50% of this body is shared by an alien set of cells. Our relatives though, right? Now, bacteria they're everywhere around us. They're extremely diverse. Now, what do you associate a bacterium with? Often, whenever we talk of bacteria, what do you associate it with?
An infection.
Infection, diseases. Yeah, they are a big part of life. They do cause diseases, but many bacteria are basically doing extremely important things. They are keeping us alive, actually. Without them, do you know that we couldn't even... There are some vitamins that we make in our body that are entirely made by bacteria. Of course, you may have your vitamin pill, but without them, you have bacteria that make it. Further, they are there as part of our normal flora. They ensure that our body isn't infected by other bacteria, right? So there's a normal flora in our body. Easiest way to see bacteria. Take a spit sample. Or you see this plaque in your teeth? Remove a bit of plaque. Put it on a slide, put some water, and you'll see bacteria around. Now, bacteria morphologically are often very uninspiring. When I was in microbiology class, I was very excited when I stained my first microscopic slide. And then I thought, what? Just rods and spheres? So often bacteria are just rods and spheres. But they can be really way more exciting than that. So here's a sample that I'm going to show you right now. And let's get the thing rolling. Let's get it rolling. It's a bacterium called as a Cyanobacterium. Okay, let's move this. So I've just loaded the slide. I have a culture of Cyanobacterium right there. And now I'm going to show it to you under the Foldscope. Here's my Foldscope. I have a light module. I also have a light that I stick around down here because often I feel that I can get some really good images with this light, which I turn on like this and so on. But today we'll try to see if we can use both of them. All right, so now without much ado, let's get going. Okay, so here we are. You see, this is what is called as a Cyanobacterium, right? Okay, let's move around and get a good view. Let's get a good focus too. Yeah. You see these cells? You see these cells?
Yeah.
What do they look like?
Veins?
Little chains. Do you see the chains of cells?
Yeah.
Right? And if you look very carefully, you might find one cell that is way bigger than the other in the chain. Let's see if you can find any of those.
Oh, right here.
You do see it?
I see some larger ones right there.
Okay. So, this is a very important organism called nostoc. It's a cyanobacterium. The great thing is, you've read about photosynthesis, right? This can, in fact, much of the photosynthesis on Earth is done by bacteria, believe it or not. And here you have a cyanobacterium, and often some of these cells also fix nitrogen. These are very special cells. Now you see that, let's get a good view. All right, so here you see a chain. Right here you see a chain of cells, and you see that one cell that is not necessarily green but slightly round in the middle there.
Yes.
Right there, right about there, right? Now that cell does not photosynthesize. It is a specialized cell, and what it does is, you know how much nitrogen is there in our atmosphere?
How much?
About 70%. And we need these bacteria to get that nitrogen down onto Earth into an organic form that you and I can consume and eat. So nitrogen fixation is one of the most important functions that are done by a variety of bacteria. And plants and animals and all the rest of us also use these organic compounds, organic nitrogen that's fixed for our food. So now the interesting thing about cyanobacteria and these bacteria is the following. Notice that often when you think of bacteria, you think of them as single cells, right? Everybody says, oh, they're unicellular forms and the like. But in reality, bacteria are often multicellular. And what we see is that a lot of the principles of multicellularity that we have learned in eukaryotes, right, like you and me, also are there in bacteria. I just showed you this bacterium with one specialized cells. But there are other bacteria that have fruiting bodies, that have extraordinary migration patterns, extraordinary signaling patterns. They go apart and come together. They hunt as a group. The diversity of bacteria is tremendous. The only thing is that when you look at bacteria, they just look like little circles and often like rods. But when you start applying the principles of science to them, they get extraordinarily complex. Okay, so that's so much so for Nostoc. Now, after a long time, I have a very special bacterium that I have found. Another Cyanobacterium. And just hold it. Be there. Let me quickly make the slide. And this is going to be a big surprise. And by the way, if you are going to be there till the end of this cast, this video cast, this live workshop, you are going to see a very special guest, very special to Foldscope. I'm not going to tell you who that is or what it is, but it is a very special guest. Okay, so there I am. This is my next sample. Let's see if I can get it in. Okay, not bad. Let me just focus it a bit. Okay. Okay, now let's turn that around. Beautiful. Let's go on to the Foldscope. Let's pin it on. Okay, let's put this here. And now let's watch this. Look at this sample. This is an amazing Cyanobacterium.
What is it called?
It's another Cyanobacterium. You just saw something in chains. Now look at this one.
These are more like cubes.
More like cubes. And how fascinating is that? This is a multicellular bacterium too. You see it?
Yes.
And do you see the cells, how they're arranged?
Yes, those little circles.
Yeah, they have a fascinating name too. I think they're called Paramecium. They're found in ponds. And once you get them, I'll tell you all what you could do as to maintain cultures. That's one of the fun things I love to do, is to maintain cultures. Okay, this is actually very easy to maintain. All you need to do is, once you find a Merismopodia, you take it from your slide and then you put it into any kind of container, and then you put some soil in it. They generally tend to grow in it. Of course, there are more techniques. Oh, now look at this. This looks like a circuit, doesn't it?
Yeah.
This looks like an electronic circuit. Wow, look at this. Now look at this. How fascinating is this?
They're so beautiful. And
Yeah,
cool.
these are cyanobacteria too, but they don't make those heterocysts, those special cells. But they might have something special. But when I look at this, the first thing that comes to my mind is, how do these things divide? How do they ensure that they are flat like this? Why, or a cube form like this? And the next question that often comes to mind for a biologist is, why is it so? Why does it have the shape? Why does it aggregate like this? The funny thing is, there are many problems in biology that haven't been studied because professional biologists, they have their own favorite systems. They have genetics in certain systems, and you cannot study everything. But what if a million Foldscopers with some pre-planned idea look at some things, maybe study Merismopodia, who knows, right? So you can have many more hands just by increasing the number of people who have access to microscopy. And that is why I love the Foldscope. It is independent of what you do, what you study, whether you're a biologist or otherwise, whether you're an artist. You have your own way of looking at the microscopic world. And who knows, you might make a discovery that might help the rest of us. Okay, so this Merismopodia, brilliant. Wow, look at this. They seem to be in a gelatinous sheet of some kind. All right, we can look at this forever, but we don't have much time. We need to move on, correct?
Yes.
All right. So now let's move on to something else. And perhaps if you hold up the phone and okay. Now, here's an interesting thing. Why don't you just focus it down so it's easy for both of us, right? All right. Here you go. We spoke about archaea and bacteria. Archaea, bacteria, right? And then the eukaryotes formed by the fusion of an archaeon plus a bacterium give rise to the eukaryotes. What? Right. Now the eukaryotes are extraordinary. The thing is that people have looked very hard. They try to find organisms that do not have an archaea or a bacterial component. They are trying very hard to find so-called proto-eukaryotes without either an archaeal component or a bacterial component. But no, so far this hasn't been possible because the defining event of eukaryogenesis, as it is called, is the fusion of these things. Only after this, a whole set of new things came about. And let's look at, now from the basic tree of bacteria and archaea, here's a small lesson. Here's something that you all need to remember. Tree of life, bacteria and archaea. Bacteria radiated into many different forms. Archaea radiated into many different forms. We saw a form today, the Cyanobacterium. You take your spit, you're likely to see 200 species of bacteria that inhabit your mouth. There's a very specific type of archaeon and a very specific type of bacterium called as an alpha proteobacterium that fuse together. What this means is that the many bacterial types already formed before the formation of the eukaryotes. How cool is that?
Amazing.
Amazing, isn't it? So now let's go to the eukaryotes, cells like us. All right? So now, what the thing is, for a biologist, it's always about comparing. You start comparing things and try to put them together and ask the question, how are they related to each other? Right? So often, this is not a complete process. It's constantly evolving. Even today, you'll find a publication in a very prestigious journal talking about solving the origins of the eukaryotes or solving the diversification or the phylogeny of eukaryotes. So now let's look at this. Okay, perhaps I should put it down here. All right, so we have the eukaryotes that formed as a result of this fusion, and there are various branches that came out. One of these branches, a very basal branch, is a parasite called Giardia. Another one, something called Trichomonas. Many people group these together. Then you have something called as Euglena. You have kinetoplastids, and so on and so forth. And of course, I will not be able to show you all these branches of life today. We don't have time, and I haven't yet come to where we are, right, on the tree. So you have Giardia, Trichomonas, I'll show you that. And I just left it out here. And after 2-3 months, when everybody was screaming at me about my bottles, I went and looked inside, and there was this extraordinary ciliate called as Paramecium bursaria. I still have it. This has been going on. I mean, let's look at the date. Did I have a date on this? No. But this is over 6 months old. And this is survived this long. Why is that? We'll find out shortly. So that is one. Another extremely long-lived culture I have. You all know how to use a Foldscope. So we shall put that in. Okay. Okay. And sometimes can be a little bit of a struggle, but make sure that you have your sample there, and then the light module. Turn it on. Take a look. Okay. Maybe I should turn on this. The thing with liquid samples is that it's often a hit and miss. You get a liquid sample, you might get something interesting, and then they just run away before you show it to anybody. And now I'm going to show this to I don't know how many people. Okay, here you go. I found a very interesting sample. Here you go. Now let's use this light and see how it goes. Can you turn the camera around? Okay. So, turn it around. Now turn it around this way, right? Sorry. Turn that around. Okay. So, here you go. Here's my light source. This is another way I love to look at samples. It's just a regular light, but it's a gooseneck lamp, and I just have it hanging in a way so that I can look at it quite easily. Alright, so now let's look at this. And let's look at this sample. Ciliates have fascinated people for hundreds of years. They're extraordinary. It's just that, when I look at a ciliate, I can't stop just, staring at it and not talking for some time, because I'm sort of interested in knowing what can I learn by looking at them. Ciliates are so called because they have cilia. And these cilia are the ones that beat around. They're found all over the ciliate, by which they move around. Now here, let's see. I need to do some adjustments here to make sure that we have, there you go. They're really moving around fast. And some might be stuck. Okay. Let's get them. Okay. There you go. Now this is a ciliate called Paramecium multinuclearium. Moves really fast, and doesn't like us, or is very shy perhaps, right? And now see, oh wow, just caught him. There is another ciliate here though that I want to draw your attention to. Okay, that's Paramecium. That's Paramecium for you. Wow, look at him diving up and down in the slide. Okay, so now there's another ciliate that I have put together for you. There you go. There he is. He doesn't want to move very much. This is Paramecium bursaria. He's kind of stuck there. What do you see inside him?
It looks green.
It looks green. You know what it has done?
What?
It has eaten a bunch of plants called chlorophytes. And you know what? It doesn't need to look around for food. All it needs is to use the photosynthetic product of the alga that it has swallowed, and in return it protects it. So here you have two ciliates of the same species. One Paramecium multinuclearium and the other Paramecium bursaria. Paramecium bursaria love light. They're extremely easy to grow. Go to any pond and just let it be for a few days. Make sure you have a good light source and you're likely to get them. Wow, how cool is this?
Amazing.
Can we zoom in a bit? Can we try to zoom this in a bit?
Sure.
Can you help me here?
Okay.
Here you can see the chloroplasts. They're not chloroplasts. They are algal cells okay, it doesn't zoom in anymore. Oh, it does. There are algal cells that are moving around. You see that?
Yeah.
Just like some people like to call it chlorosis, but it's just the algal cells moving around as our Paramecium bursaria poses for us so that we get a wonderful view of it. All right. So much so for ciliates. But notice one thing. People often think that humans were the first one to start farming. Nah. You know, ants farm. That is also not surprising anymore. But there are these single cells that farm. Now, how cool is that?
That's amazing.
Just imagine you have plants growing inside you, so you never have to eat. How much time will that save you over your lunch break?
At least 30 minutes.
At least 30 minutes indeed. Okay, wonderful. Well, we have to move on, my friend, because we have other people waiting for us. So let's move on. Now, here, tree of life is to actually understand the different stories of these organisms. Often these stories are not told, or were told 100 years ago and forgotten. Now, there is another ciliate that is my absolute favorite. Do you know what that is?
Which one?
It's called Tetrahymena Borax. There are stories that still need to be told about organisms, and this is possible. The great thing about the Foldscope is that you don't need to be in a big lab in a very wealthy place with all kinds of resources to look at this. You can do this. You just need a little bench, a light source, and your Foldscope. And the world of magic is in front of you, and you can do some really interesting experiments. So now let's look at this guy, okay? This is a ciliate called Tetrahymena Borax. You know it's my favorite. The reason is I've had this now for 3 years. I've been growing this and I've never had a problem. It grows very easily. I've always enjoyed working with Tetrahymena Borax. In fact, whenever I want the oohs and ahs from kids or from people, I take this along with me. And he's fantastic. I don't know, he, she, it, no idea. So now let's look at this a bit. As always, we just have to ensure that sample is there. Right? Just have to make sure that you have a good sample out there. All right. So, there you are. I've caught you in the act. All right. Now let's look at my favorite ciliate, Tetrahymena Borax. Let's get this here. Okay, you don't need to change it. You don't need to change it. Okay, you don't need to turn this around because it's already turned around. All right, so now let's put this here. Let's put this down here. There you go. I'm going to use the same light source as before. Now, this is a remarkable ciliate. I cannot tell you how happy I am to see that. I actually saw them after 6 months and they're still doing reasonably well in my hands. I may have to passage them in the next couple of days. Notice the thing about the ciliate is that it comes in many different shapes. Now, I do not know if the many different shapes are seen here. You definitely see this shape, right? There you go. You see that big guy there? You see this guy there? He is... Okay, see this. Let's get it. Oh, you see that one?
Yeah.
No, that one with the big open mouth that we're chasing? That's a cannibal. Wow, you know what a cannibal is?
Well, an organism that eats another one of the same type.
Yeah, and you know what? How this survives? This is a extraordinary, remarkable thing. I wish I had learned this in school. I would have become a tetrahymena biologist. Look, what happens is when there's less food in the medium, some cells become giant like this, and they just become big open mouths, like you see here, right? And then these big open mouths, they swallow little guys, right? They get their food, then they divide and become small again. And you know what? It's fascinating to watch these. They can be much. This one is actually a mini giant. There are ones that can be 3-4 times bigger than these other cells that you see there. Now, look at this one. This one's dividing. Wow, look at this.
Whoa.
How cool is that?
They're like twins.
Yeah, they're about to divide. There's still some food around. So what happens is that there are 3 forms of this ciliate. One is this one is the one with the giant mouth, and then there is one with a tail. Let's see if we can find the one with the tail. So these 3 are actually in a rock, paper, scissors game. You remember the rock, paper, scissors game that we often play with together?
Yes.
What is it?
Well, each different player, as in rock, paper, scissors, can defeat another one, but only certain types can do that.
Okay, so give me an example. Rock.
Like rock crashes scissors.
Okay.
And scissors cuts paper.
Okay.
And paper covers rock.
Okay, so none of them actually win, and that's what's happening here, I think. Now, that's my hypothesis. The beauty of biology is that when you start seeing interesting things, you need to form hypotheses in your head. Why is this? What is this? What might be happening? You may be absolutely wrong, but you know what? There's nothing like testing a good idea. So what I think is happening is a rock, paper, scissors like situation. See that giant? Wow. So oh wow. So the giant one eats the smaller ones, right? Now these tailed ones that I don't see here. How do you turn this around? Okay. These tailed ones, they are not, these tailed ones are never eaten by the giant. But the tailed ones don't grow as fast as the other ones. So essentially, they are all competing for resources, and they have different strategies to win over their resource or to win over the other. But nevertheless, because it's a rock, paper, scissors type of thing, they are always together. You always find all the 3 forms at any given point of time. How interesting is that?
That's so cool.
Would you like to have this in your elementary school? Would you like to show this to your teacher?
Yeah.
That will be super, isn't it? And such an interesting problem. Now, the thing is, I have an unusual hypothesis, and I think I just had it today. And that is that, this has lasted for 6 months. I didn't give it any food. I didn't do any. They eat the little ones, they divide. So they are sort of trying to be in a self-limiting cycle. And perhaps because of that self-limiting cycle, they're able to live really long. Now that's a hypothesis. This can be tested another day. Let's move on in the tree of life. So where are we? All right, so here we are. Ciliates. Ciliates. There are hundreds of types of ciliates, and thousands actually. I have this book, The Illustrated Guide to the Protozoa. You can be looking at this book all day and reading about all kinds of interesting things about different things that we call ciliates. But you know what? The greatest fun is when you have your own collection of ciliates. And how do you collect these ciliates? How do you make a collection? And that's what I'm going to focus on in the next 5 minutes. It's very easy. You just have to try a bunch of things. One of the things I love doing is the following. Take an empty bottle like this. All right. I always have There are a couple of things that a person who grows things at home needs to have. A good pressure cooker. Some bottles. Good friends, because, mom can give us this bottle, but there are some really good friends who can give us something like this, right?
A beaker.
A beaker, absolutely right. Or sometimes you get them in specials in stores. Take a bottle. Another thing that I love having, boiled wheat grains. Extraordinarily useful things. Take some wheat, put it in a bottle like this. All you have to do is, so let's see if I have, there you go. I have some water here. I put this in this pressure cooker, so it's supposedly sterile, free from organisms, because things tend to grow very rapidly if you just let them be. Just water, just plain good old water. Most of us who try to grow things, we prefer to use spring water. You get plenty of this nowadays, but essentially you can take a water body, heat it up, or leave it aside for 24 hours. If there's chlorine and other such chemicals, they will go away, and then you have this. Usually, I love to use my Bunsen burner. I don't think I'll turn it on today. That's too much of a show. What you do is take a couple of grains, dump it in. In reality, I have just used 2 grains to grow things. If you look at my cultures, let me show you some things that I might have. There you go. Look at this. Here's a culture of something I've been growing. No name. I don't know what it is yet. But you see these little wheat grains and water?
Yes.
You leave your sample in that for a few days. Leave it in it for a few days. Maybe a week, maybe 2 weeks. And magic happens. Look at it every day if you can. Look at it every 2 days if possible. And you will see a variety of life forms coming up, right? So you have this, but you have to put something in this. Now, in my case, if I have Paramecium that I need to grow, I don't know where I put that. Put it back, I suppose. Where did I put it? Somewhere here, right? Okay. I have something else. Oh yeah, here it is. Paramecium multinuclear. That's it. Let it be. This is how you maintain cultures. Very easy. Wheat grains and plain water. Now, there are some organisms that you don't even need to go that far. Look here, I have so many bottles. This is just pond water. Good old pond water. Stuff growing in it, which is great fun. Yet other times, let me see. Just be right there. Okay? So I have this piece of lichen. See this? This piece of lichen. I just take this lichen and put it in water. Here, I have a plate. This plate has been used 100 times. It's still okay. It's a plastic plate. There's some lichen. I put it in water. And the forms of life that you can see is endless entertainment for days, months sometimes. All right. So this is a very easy way to grow things. Another easy way to grow something called as the hair infusion. Take some hair. Take some hair, right? Cut them into small pieces. Put it in a bottle again. Boil the water for 10 minutes. Put it into a microwave. Put it in for 10 minutes. And then into that when it cools down, you can put some soil, you can put pond water. There are many posts on Microcosmos on the Hair Infusion. You guys should be going there and looking at some of these posts. And they're fantastic. Okay, now, we still have some time, I think. And I think I'll continue a little bit more. All right. All right, so now let's move on, right? I've spoken enough. Hair infusion, boiled wheat grains, 2 wheat grains for about 20 ml. That's all you need. Magic. Now I'm going to label this because I might forget. Usually, put this on top. Paramecium, Multinuclearium. What is today's date? 6 June 2020. Okay? And to hide it from everybody, I put it in my special place. Just keep it in a dark place so that nobody messes around with it. Alright? Alright, so now let's move on. This is how you grow a variety of life. And you'll be surprised the kinds of things that grow in them. There's not much time remaining, so let's move on along the tree of life. Okay? Essentially, what are we doing right now? We are looking at branches from a major branch. You're seeing a large number of species up in the sky. So think of it that way. All right. So we have looked at ciliates. So we've looked at this part of the tree of life. I have something from this part of the tree of life that I will show you. And if we move along, there's another group. Very famous. Plants. Do you know something? Plants, essentially, a plant is a cell in which it captured a cyanobacterium. It did the same thing that Paramecium bursaria did. But unlike Paramecium bursaria, this cell was tremendously successful. Not only did it diversify into various forms, it actually went on to have the great vegetation around us, the trees and the large number of eukaryotic algae. They're a very important aspect of the Earth's cycle. So I'm not going to show you much about plants. If you were there in the beginning, I showed you a spiderwort, the trichome of the spiderwort. And I think I'll stop there for the plant because many of you have already looked at plants, onion peels and so on. The Microcosmos is a wonderful resource. Just go there and look for. Now let's go to this branch. This branch has got 3 interesting organisms that are very prominent. So let's just say I rub all this out and only focus on the last branch. Okay, so this is it. Important. It's very important because the tree of life helps you make good biological hypotheses. There's a very famous biologist, Theodosius Dobzhansky. Dobzhansky said that nothing in biology makes sense. Nothing in biology makes sense except in the light of evolution. The tree of life essentially tells us that life has evolved over a large amount of time into these different forms. And at different branches, why do you have something? Is something that fascinates the biologist. He wants to know, okay, plants are green, but why? Okay, they got the chloroplast, but why? When? Such kinds of questions can only be answered. Let me see. Let me ask. Maybe Rebecca will send me a message. Okay, that's fine. I'll move on. I'll go on. Okay, we'll stop when we are done. How about that? You agree or no?
Yes.
Pieces that you see folded really well, and then you're generally good to go. Right? Okay, so that should be fine. So now let's try to find something interesting. Okay, let's see. These are a bit Once you get them, they're easy, but they can be a bit of a pain. Let me see if I can find a few amoebae. Now I grow amoebae. Amoebae grow very easily. Again, a couple of wheat grains, amoeba. Sometimes they like other little organisms called ciliates, other ciliates. One such ciliate is Chilomonas. You can culture an amoeba for years with Chilomonas. Okay, then let's try. Let me try to find an amoeba or two here. The thing with amoeba is that they are transparent. So you need to be you need to be able to look around very carefully. Okay. Is this my amoeba or is this something else? Says amoeba. Nah, this is not an amoeba. That's what I thought. Borax. Oh, here it is. He's right here. Sorry. So let's look at this without much waste of time. All right. There you go. Nice. The thing that I cannot do right now is patiently go over my slide. I don't like to be in a hurry in reality. Normally, I spend the whole of Saturday doing this. After lunch, nobody disturbs me until it's dinner time. Microscopy is a slow process. Do not think of wanting to get some penny days. Ah, there you go. Remarkable. Remarkable. Nothing like watching an amoeba. And I'm going to show you a very interesting trick. A light trick with amoebae. Okay, so now let's put this on. Do we have enough charge?
Yes.
Okay. Okay, let's, where is the, right here? All right. Okay. So now again, I'm just using the regular, light source. And there you go. What do you think that is?
I don't know.
Okay, nor do I. Let's just make sure that this, how do you zoom in? Okay, thought I had him there. So you see these little things that are floating around? This is it's not, I think it's a ciliate. It's one of those things that you just give. You don't think about what species it is, but it is likely to be a ciliate. It's called Chilomonas. And it is usually used to feed amoeba. And I thought I saw a few good samples. Let's just look around a bit. Okay. Ah, there you go. See this? This is what has fascinated people for ages. This is your amoeba. How cool is this?
Amazing.
Amazing. And now let me show you a trick. I'm going to show you a trick that you can actually enhance amoeba and make it look really cool. Okay. Watch. Come here. Look at this background. What's happening to it?
It's become red.
Yeah, that's a cool trick, isn't it? Now here's another one. What is happening to it?
Now it's turned blue.
It's turning blue. Now if I combine the two of them, I might get an interesting effect.
I'm thinking it might become purple.
Now, look here. Look carefully. What's the background now?
No, it's The ciliate is red.
No, ciliate is red. What about the amoeba in the center? What color is this?
Like a light pink.
Light pink. And what is the background color?
Blue.
Right. Look, how did I achieve this? This is one of the funnest thing you can do with the Foldscope. It's called Reinberg illumination. Okay, I know that, many people have only been there for 1 hour, but nevertheless, let's just look at this for a second, right? Look at this amoeba, how amazing it is. The thing is that a lot of life does not have very great diversity in morphology. And a lot of life forms, they just look like this. In fact, there are some organisms that are very close to animals that just look like this, but are, very close to the animals compared to the Amiibo zone. And wow, let's just stare at it for a second. You can see cytoplasmic processes in them. If you are lucky today, but that might take a lot of it's actually hunting for chilomas. We might get one capture. Okay, so I'm giving myself another maybe 10 seconds. You can see the streaming. How beautiful is that? How beautiful is the streaming? Amazing. Okay. Well, I can go on, but this is only a workshop. But you know what? Once you get an amoeba, you don't let it go. I'm going to hang on to this slide till after the workshop, and I'm going to be looking at this forever. Now, what did I do? How did I get this special effect? It's very trivial. It's very trivial. So what you do is, here's your Foldscope, right? There's no light module here. And now what I have is gel paper. It's just good old gel paper. It's transparent. Can you hold this for a second? Thank you, dear. Yeah. Thank you, doctor. It's just plain old gel paper. Red and blue. I just put it in here, right? And then put my Foldscope there. And actually, I don't need to use my phone. Oh, wow, I see the same thing. It's just remarkable. You get a very special effect. The advantage of this is if something is transparent, then you get to see its foreground and background, and it's amazing. All right. Now, we can go on and on. Okay, but maybe I want to show you something special. I don't seem to find it. But I love collecting slime molds. These are Amiibo Zones. What happens is, these amoebae, they come together when there's starvation and they form fruiting bodies. They are found all around Northeast America. And they are very fun to collect, very fun to grow. I don't know where I put mine. Maybe I'll try to get it at the end of the workshop, if possible. Okay. There's a lot more I can show you all. But let's go along the tree of life and keep it simple. Fungi, you've seen mushrooms. Just put some bread out and you're likely to get rhizopus or some such, or mucor or some such thing. And then your yeast cells. I have a very famous mycologist in India. I think his name was Mani. He was a remarkable mycologist, and some of his works he has basically studied fungi across India, and some of his works are great. Maybe what I should do is I should put it up on Microcosmos for everybody to study. He's one of the doyens of mycology. He has discovered so many interesting species of fungi. And yes, he had his microscope, but I can imagine that if he had his Foldscope, he would be really excited. Okay, so let's move on. Finally, vertebrates, insects, plants, and fishes, and so on. Vertebrates and various invertebrates like ants and insects. There is a very interesting animal that Manu works on. It's called Trichoplax. It's extraordinary. It's just a sheet of cells. That's an animal too. Okay. Without much ado, and I think I'm sorry now. All right. So let's try to find him. I found this in a lichen sample with great difficulty. I mean, I may have made something like 30 slides just looking. I thought, okay, I'll get him. And finally, I thought I saw him. He actually grows. This thing is a remarkable species called as a tardigrade. Now, the thing about lichens, look, here are all my lichen samples. The thing about lichens is that, look here, here's a lichen sample that I just got from outside. These undergo drying and wetting cycles constantly. So anything that grows on them is adapted to this drying and wetting cycles. So for example, if you grow some critters in this, after some time, they just eat as much as they need, and then they immediately go down and form spores. Colpedium, for example, a kidney-shaped ciliate, grows in them, and it will grow very well for about 10 days, and then it will form all these spores everywhere. Similarly, nematodes, rotifers. I have a rotifer too, but we don't have time. And of course, the tardigrade. Okay, now let's try to see if we can find them here. The thing about tardigrades, and they'll be fine. Some of them are even resistant to ionizing radiations. So all kinds of studies about for ourselves. Let's just see. Okay, I'm going to give myself another 10 seconds. And lo and behold, who is there but our good old friend? He wants to say goodbye too. So let's put him there. Put the camera there. Put it there. Oh no, like this. This. Put this on top of this. And there you go. There's nothing like watching a water bear in action. Maybe zoom out a bit. It's a remarkable animal. It often forms eggs when it exists. And you see those eggs in there, and you can see eggs develop. This, by the way, is the mascot of the Foldscope project. In a sense, many of us start our journey looking for things like a tardigrade. You won't believe that when I was younger, much younger, I wanted to see a tardigrade, and I started looking for them, and I didn't find them. But in the process, I found so many other interesting things and learned about so many different things. That's the advantage of microscopy. That's the advantage of exploratory work, exploratory research. You come across a variety. You may have a certain goal, but in the process of achieving that goal, you learn so much about other life forms, other organisms, and about their biology, about their shapes, about their life cycles. Sometimes you may find things that nobody has written about. And that is when you can have that good smile to yourself. Hey, I know something that most people in this world are unlikely to know. And that is a big motivation for many biologists, is to find that unusual thing. that insight, that is true for that organism, and it also forms a little cog in our understanding of this universe of life, that is this tree of life. Trying to fit a small point into the tree of life is as wonderful as actually looking at the tree of life in its entirety. Okay, I think with our friend here, I am going to stop. Let's take one last look at him. This one grows in lichens. Normally, if you want to get a tardigrade, for every 5 lichen samples, you're likely to get a couple of tardigrades. That's how it is. I just have to go out. I don't have to walk very far from my place, and I usually find them, in lichens, and I keep the lichen aside like I just did, in case I'm bored and I want to look at them again. Okay. So, just one last part. Are there any questions? Yeah. Well, I think, there's only that much you can do in one life. There's only that much you can do in one workshop too. But there's something else I want to share with you all, right? This is towards the end. The Foldscope is a remarkable project. It has changed the way I look at biological systems. I'm a professional biologist, but the thing is that the Foldscope has changed my own attitude, not only towards biology. Yes, biology, I'm a professional biologist, but also towards sharing of things with others. I never knew how much joy I get sharing my discoveries and my findings with kids. I actually started a club called the DC Micronauts. It was just a bunch of kids. I said, okay, let's go and explore. We did workshops. We tried to do some exploratory work. It was fantastic. I felt very fulfilled. I meet a lot of extraordinary people, a lot of extraordinary scientists. But the best thing I like about kids is their curiosity, the questions they ask. It's the same questions that they will ask in a different way when they grow up to be engineers, scientists, bankers, whatever. But that innocence in that question is what we need to have when we do our work. Also, in terms of the scientists, the best thing is, I told you that there are about 10 million to 100 million species on Earth. Somebody estimated that it would take about 500 years for us to actually catalog each of these species. Think about it. And that's only professional biologists. Now, if we have 1 million Foldscope users trying to look at different species, cataloging them in a very systematic way, we might be able to catalog a great number of species in our own lifetime. The second thing is, 90% of life is microscopic. The diversity of microbial life, microscopic life, is way more than what you see around us. For example, recently we found that multicellular bacteria are likely to have something like apoptosis, where some cells die to save the others. It's predicted theoretically. It should be there. We haven't found it yet. So go there. Take your Foldscopes, explore the world. One last thing, books. We all love our books, and I love what Manu did in the first workshop, sharing some of his favorite books. There is no one book, but this is one of my favorite books. This is For the Love of Insects. I've read it so many times that the cover is gone. It's written by this biologist called Tom Eisner, and he talks about chemical ecology and the diversity of interactions of insects. And it's a fantastic story where he combines chemical insights into biological insights. The great biologist E.O. Wilson said, and now I recollect, that if I were to start my career again, I will be a microbial ecologist. Think about it. With a Foldscope in hand, you have already started on this journey. There is so much that we don't know, and so much that we can do right from our benches. That is one. Another book. I love this book. The Marvelous Animals, written by Helena Curtis. she talks about different life forms in a very easy to read way. I must have read this book at least 5 times. It talks about microbial life. There was a time when people thought these were animals. But no, once we got better at looking at molecular data, we are able to form this thing called as the tree of life. There are some other books I have, but I think I'll save that for another workshop. All right then, friends, fellow Foldscopers, I hope you are going to go back to your Foldscope and enjoy your journey. There's so much to learn, so much to do, and it's never-ending entertainment. Thank you. And now for my little helper, Dr. AI. Let's hear a goodbye from her.
Goodbye.
Goodbye. Thank you all. Okay, so now we need to stop this. How does it stop? Okay. That's it.